Why are some microbes resistant to treatment and others not?
Hi, I'm Claire Reichardt, and my research focuses on the microbe Cryptococcus neoformans, which is a human fungal pathogen.
Cryptococcus commonly resides in the environment, growing on trees. However, when inhaled by a human, it can travel into the lungs and eventually disperse into the brain in patients with weakened immune systems. Cryptococcus neoformans is estimated to cause 147,000 deaths annually. Patients who contract Cryptococcus neoformans are commonly treated with antifungal drugs. We know for Cryptococcus neoformans and many other microbes that under the same dosage of antibiotic drugs, individual infections can have varying resistance levels. Sometimes Cryptococcus neoformans can be extremely resistant to drug treatment and continue to grow, or sometimes it can be extremely sensitive to drug treatment and hardly grow at all, and sometimes it's somewhere in between. This is common between various strains of Cryptococcus neoformans, which we also know have different genetic backgrounds.
Therefore, the main hypothesis of my research is that different strains of Cryptococcus neoformans have different levels of drug resistance because of their varying genetic backgrounds.
To test this hypothesis, I will have two approaches. The first approach is for me to analyze the genomic information of Cryptococcus neoformans strains from around the world. To do this, I'm using the whole genome sequences of 384 diverse Cryptococcus neoformans strains to create what is called a pangenome. A pangenome is simply a collection of all the genes from a population of one species. The pangenome I have created will be used as a way for me to capture all genomic information about the Cryptococcus neoformans strains. The second approach I am using to confirm my hypothesis is to test regions of Cryptococcus neoformans genomes. To do this, I will create a population of mutant cells using a technique called TNSeq. Simply, this method uses transposons, or mobile pieces of DNA, to inactivate regions of a genome, creating millions of mutations. Then, I will expose this mutant pool to a selective pressure and antifungal drug. The result of this technique will be mutants that are resistant to the antifungal drug I have exposed them to. This will help me discover regions of the genome that are important for antifungal drug resistance. The broader applications of my findings are for use in diagnostic procedures for patients. Knowing which genes are correlated with drug resistance will help decide which antifungal drugs to provide for patients based on the strain they are infected with.
Cryptococcus neoformans is already extremely difficult to treat. Choosing an effective antifungal drug will help accelerate the treatment of infected patients as well as lower the overall mortality rate. I would like to thank the ARCS Foundation for selecting me as a finalist, as well as my lab for all of their support.